there is some kind of there is a learning required. So what you all have to do now? Now, if you see about this, most of these are a plant products. So once you will open it up, any one of this, I'm not taking specific examples, so then you all will do, but I'll just give you outline how to make this assignment. Yeah, and these are going to be a definite question. It can be about 8 to 10 mark questions. So what you have to do is, which are the plants through, what are this medicine? In the first place, your introduction to the topic should be, what are this medicine? Who are the discoverer? Which are the companies which have really discovered it? What it is used for? Then all of them have some relation to plant or some chemical, certain natural synthesis. So you should write about that plant, where you can collect, because you have understood sample collection is a bioprospecting, and that is how we will progress. Correct? Any doubts you have, any one of you, so that if that doubt will be whole class doubts, that I can clear it, so that you come up with a good this, what do you call, your assignment, and these assignments then we will share with each other, three groups. So these topics I'm not going to teach in a class, because this is self learning as per the college requirement.
So,
Yes.
so sir, do we need to write the whole process of the formation of drug or just till the sample collection?
No, whole thing. Okay, I stopped at that. So you will collect the plant, how these plants were processed to get this. So you'll have to do a lot of reference work. Not one. If you go to only, what do you call, our Google search, use Google. I'm though most of the teachers might be telling you don't use Google. I'm fine. But Wikipedia, don't only just rely on Wikipedia. You might have to read a small paper. I'm not expecting at your level to go into a very complicated paper. But like, if that is like RT News or RT Missing, so once you read about it, then if I'm just taking examples, it become more clear to you. That artemisinin is anti-malarial drug. So what is the plant use? So which are the malaria is caused by which organism? That you can write. So all those things you have to write, and then you have to write about the action, mode of action of this drug. Then you have to write about the toxicity part of it. Yeah. So this is how it will become three full scale. Did you understood, Shakti?
Yes, sir.
Okay. Anybody else has any doubt? Because from next class onwards, we are going into little basics thing, because now paper setting is going on, and I have seen some of the paper setting process and some of the paper. So we'll also learn more from examination point of view, and lot of your syllabus is still left. So we will go into a basics of not just bioprospecting application part, but we'll go into a fundamental part. Yeah, fundamental is also very important. Some of you want to pursue science and don't want to go for work immediately, or go for a PhD in a long range they would like to have. But still coming back to this assignment. Yeah. So even when I go to anti-cancer drug, that paclitaxel, Taxol was a wonder drug. It came from a plant. It's a plant bark. Bark was used for this. So you find out about that plant and write about it. Then it was used for which kind of cancers? What is its mode of action of Taxol? It was like cytotoxic drug. How it is tubules related. So you'll know about something about that cancer. It was used in a breast cancer. So you will write some small paragraph. I'm not telling you become expert in the particular cancer, but using this particular route of pharmacognosy, your knowledge of drug, disease, and the plants, they will improve. You will understand using this example, and then we will progress further. Okay?
Yes, sir.
Okay. So I'll still wait for another 3 minutes till 35. कुछ क्वेश्चन है तो पूछ लो, फिर हम नया चीज स्टार्ट करते हैं आज.
Excuse me, sir.
Yes, sure.
So I have a question. So, sir, I'll be writing about photophyllotoxin.
Can
So,
you,
in this,
can you start your camera if no problem, if you have no issue. Otherwise, if there is a bandwidth issue, don't start.
uh, there's like a bit of problem because of
ठीक है,
that.
ठीक है. Okay, go ahead. Ask your
So,
question.
so basically, in this talk, do we have to write only till its procedure on toxins, or do we have to write other toxins or drugs that are like
Huh.
derivatives of this?
Hi, yeah. So see, if you have a podophytotoxin and miltefosine, these are the two different compounds, correct? I deliberately gave because there is not so much information on miltefosine because it has been used in only third world country dishes, leishmaniasis a lot, but it was used for multiple. And podophytotoxin, that is also a little less information you will find, but this is in your syllabus and important. Now, from where you can get miltefosine, how it is synthesized, whether it is synthesized from nature, from where you get. Podophytotoxin is produced by which uh, plants? Because so first you should have a knowledge about what is miltefosine. Let's take me if you want podophytotoxin, I'll take example of podophytotoxin, and then you can. So first you have to write what is podophytotoxin, correct? What is its use? Then where it was available. Yeah. So because we are learning this under pharmacognosy, so from nature related, whether plant or natural synthesis, that is rocks, animals, from where we have got this podophytotoxin and how medically it is used. Then if you use it little larger, what are its toxicity dose? What are the toxicity of podophytotoxin, correct? Any drug which we use. So this will improve your knowledge that every drug is good up to certain dose. At a higher dose, it causes toxicity. What kind of toxicity it causes?
All right, thank you, sir.
हाँ नंदिनी clear हुआ?
Yes, sir.
Okay.
Thank you.
So, we can keep on asking the questions on this, and I hope that I normally, I give a lot of time. Last time you all agreed for 15 days. I think I made it 16 or 17 days. So give me, but give me sharp. Then I don't really entertain a lot delay. Yeah. So please, if I am giving you it in a right time, explaining it in a right time, I expect the similar treatment other way around, right? So please give me this by 15th or 16th, because after that we will have one slide presentation from you all. That is on some other topic we will keep. So, these are your two mark scoring area. And, I've seen the students able to score good marks during these assignments and the presentation. And then class test. Class test, I cannot assure you that whether you will score good or not. That is how you do. And these are the three main before your real final exam, right? You all know about it. But I think, now you are approaching towards the final year, so your score will be very important. So I'm sure you will be very sensitive about it. So today, what I want to touch upon our bioprospecting, as I told you, applied. Next time onwards, we will change the gear. Huh? Oh, you might be seeing my this desktop. Okay, one second. Just give me a moment. So, I've sent you all two slides today. One on fermentation, and now from plant related thing today, what we discussed, you all are going to work, and at the last class we did. This is the last leg on our bioprospecting, before you forget the bioprospecting, because it's very important from your all aspect. And that is why this is not a bioprospecting. There is separate topic in your syllabus called as fermentation antibiotic. But it is at the end of it, it forms a bioprospecting only, because most of the fermentation product came from either bacteria, fungi, or yeast. And all those three products are come through bioprospecting, that is collection of soil sample, enriching the soil sample, isolating the organism, making a drug bank, using a metagenomics, find out the gene, make again the culture bank, then screen the culture bank for antibiotic. Now we are talking about antibiotic production. So antibiotic, a selective, the antibiotic which you need. And then you go for a fermentation, which we learned very well last class. Many class we learned fermenter construction, then the media selection, then we learned about downstream processing. I'm just revising this. Please be with me. I know some of you might be finding it boring, but I think it is better to revise so you get a flow back, correct? And that is why I'm doing this. Once our fermentation is over, we monitor all the parameter in fermentation, and then we using our screening model. We check whether our product is being formed. Once the product is formed at an optimum level, which can be done using screening model, at the same time we can have different probes and we keep on taking samples from fermenter, and once it is ready, we know that now my product is sufficiently made. We will harvest it through the down port, and then we will process it, centrifugation and multiple downstream processing. I think I sent you sufficient literature. I'm sure you must have gone through. So that is what the whole thing. And now we are going to do what a specific antibiotic. There we have just talked about one antibiotic. Today we will take a specific antibiotic, so it becomes completely clear in your mind that how actually the things work. So who will volunteer to read the first slide? So for me, can you all see my slides first, please?
Yes, sir.
Yes, sir.
Good. Thank you. So please start reading. I'll start explaining in between. Okay? So please, anybody volunteer. I don't want to take the names. Anybody can start.
Sir, can I read?
Sure. You need not ask, please. The first person. I am very happy that you are volunteered. Go ahead.
So, Griseofulvin is an antifungal antibiotic first isolated from a penicillium species in 1939. It is a secondary metabolite produced by the fungus Penicillium griseofulvum. The compound is insoluble in water and slightly soluble in ethanol, methanol, acetone, benzene, CHCl3, ethyl acetate, and acetic acid.
Yeah, good. Just, this one introduction is like, telling you the whole story. From exam point of view, इतना भी याद रखोगे ना, हर चीज को बाद में हम expand कर लेंगे, ठीक है? Good. I was just finishing my tea. So now I can take more easily. So, Griseofulvin, as it explained, it's a very old antibiotic, 1939, correct? Even I was not born. But why I picked up this such an ancient example? The first thing your question will come कि ये बुढ़ा इतना क्यों अपने को old चीजें सिखाता है? Old नहीं है. I'll tell you why. Deliberately I brought this slide because most of us feel that, oh, this is all research done and there is nothing much to do. Let me tell you, even in 2019 to 2020, that is 1 year, the whole Alembic company, and there are 3 other Indian companies, smaller companies, started working on Griseofulvin, antifungal. And antifungal, let me just take you to a little interesting aspect. You all know black fungus, COVID के साथ black fungus हुआ था.
Yes.
Yes.
Good. Yeah. So antifungal drugs are so important. Did you understood at that time? We didn't had a drug, and we were literally some of the hospitals were hunting for amphotericin. That was another drug, but other antifungal. So the antifungal are so okay. The black fungus is nothing but a mucor, which is causing an infection and we are not going right now into clinical part. But yes, so the antifungal drugs were very important to kill that kind of thing. And that is what in 2019-20 there was no issue of COVID. Neither this black fungus or mucor related issues were present in the world that seriously. But consistently, the point which I'm trying to make here is, this was isolated from Penicillium species in 1939. Correct? Somebody has isolated using our bioprospecting style from a soil. But just imagine that even after almost what I'll call 75 years, there was a project to improve the Griseofulvin production in a commercial level and improve the fermentation process in such a way that the toxicity because of the impurities will be less. Did you all feel that what you all are learning is really implicable into the industry and real world of real life situation. So, from exam point of view, you should remember that drysofulvin is one of the antibiotic. I'll be taking two examples. One is antibacterial and one is antifungal. So antifungal, you all understand fungi, correct? Fungi आप लोगों को पता होगा by now. So antifungal drug it was, and it was produced by penicillin grisofulvin. So this name you have to understand. Penicillium you will remember, and grisofulvin producing strain. So it is grisofulvin. Yeah. So the compound which it produces, that is grisofulvin, is not soluble in water. Now, what is the importance of this? Once you know that this is producing into the fermentation, and if you know it is not water soluble, then your downstream processing. Downstream processing is what? Recovery of the compound, correct? I hope that I think 2-3 class we spent. Do you all completely forgot about downstream processing, or you all know about it? Centrifugation, then rupturing of the cell, then column chromatography or extraction.
Yes.
Good. Yes. So, this extraction, this will be because it is a water insoluble. So we have to use organic solvents. So these are some of the very commonly used. CHCl3 is nothing but a chloroform, ethyl acetate. These are the common solvents which we use. These are organic solvents we use to dissolve and extract out from 100 or 1000 or 10,000 liter of fermenter grisofulvin into a small because downstream processing first principle is only to get it into a manageable quantity, right? So that is what this one slide is talking about: isolation, the strain that is producing organism, production, and then downstream processing. एक introduction slide में ही ले आया. Now we are going to learn the whole story. ठीक है? तो क्या हो गया? Oh shit, this is not moving. आज कुछ ना कुछ तो इधर प्रॉब्लम है जरूर. One second, I think I'll first unshare, then again share, and let me see whether it works. Oh, no. Can you see now something? Can you see my slide?
Yes, sir.
Okay, so this is a grisofulvin structure, molecular formula. Don't get scared. You don't have to remember this. Even if you just able to draw this middle ring, I think that is sufficient. And even if you can't draw this structure, you will not lose the mark. Okay. But if you draw it, I think it is going to give, after writing this introduction part, and if you draw this structure, it is going to give a great impression to the person who is checking the paper. I don't know why my slides are not moving today. Okay, can you see the another one, cultivation?
Yes, sir.
So you all have not seen the fungi, and as I told you, penicillium, correct? Penicillium is a green color fungus, and that is why this photograph tells you. And this photograph also should be able to correlate you with our screening, which we used to, isolation from a soil, thousands of organism, and then we were selecting 100 organism. Just try to remember and correlate in this class. So this is what the picture, the plate and the fungi will look like this, and then you will select the strains from here. Okay. Any questions here? If anybody has some doubt up to here. Okay, I think somebody else. So now now we are shifting our gears. So media. You remember in a fermenter vessel. In between, I'll show you a fermenter vessel also. But you remember in a fermenter vessel, we were putting a media, a carbon and a nitrogen source, which will be useful for organism to grow and produce the product. Correct? I think we did it very well. So can anybody read this medium?
Sir, may I?
Hi, yeah, please. Hey, okay.
The medium, Chesapeake, Dox medium, chemicals, glucose 5%, sodium nitrate 0.2%, potassium hydrogen phosphate 0.1%, magnesium sulfate 7H2O 0.05%.
Perfect, I think, So, as we discussed earlier also in a basics and in a fundamental way, now it is actually, you can see, for production of an grisophulvin, we use Zepax-Dox medium. And the Zepax-Dox medium carbon source, can you see? 5% of glucose, which is one of the highest. In fact, if you see, it is 10-fold of almost, 10 to 20-fold of any other component, because this is a fungi. Penicillium, I told you, we are talking about Penicillium grisophulvus, which is a fungi, and fungi need can use very large amount of carbon source. So glucose is a carbon source, yeah, and nitrate, sodium nitrate is a nitrogen source, and 0.2%. And phosphate is required for any growth. Like phosphate is a very quick, rapid growth, and these two are for production, glucose and nitrate. And magnesium as a salt for any enzymes and our DNA replication, fungus DNA replication we need. So that is why this is a very balanced media. ये मीडिया याद रखने में डिफिकल्टी तो नहीं होगी. I think this should be just 4 component. You have to write this media name when we are talking about antibiotic fermentation. So we will talk about the strain, we will talk about the isolation, one of the picture we will draw. If you can draw structure, excellent. And then you will say that the fermentation medium is comprises of this. Yeah, इसमें मैंने water नहीं लिखा है, but 0.5%, 5% means 5 gram in 100 ml, huh? 5%. So you dissolve everything in a water. In a fermentation, water is a standard media. I mean standard solvent which we use. Everything is dissolved in water, and these are all water soluble component. ठीक है, मीडिया प्रिपरेशन के ऊपर तो किसी को डाउट है?
No, sir.
Okay, so I think please read. Shakti, you only can read if you come on the camera.
Yes, sir. Steps involving the manufacturing process: fermentation, pre-treatment of fermentation broth, filtration, extraction, decolorization, isolation and separation, precipitation and purification.
I think now if you remember a little bit what we have done, the first 3 steps, yeah, are upstream processing, correct? We have learned fermentation thoroughly. Still, I will show you the fermentation, but just try to correlate with, I think we have spent sufficient number of class on that. Pre-treatment of the fermentation broth, I talked about sterilization. Then we discussed about how with jacketed vessel, we will heat it through a steam. And if you put directly steam into it, it will go into a very large volume and it will come out. That is why we first take it to 100 degree and then that's a pre-treatment of fermentation broth. So that is what you are doing is you are sterilizing it and then inoculation it. And then once the growth take place and your product is ready, you will go through filtration. And this after filtration, this is harvesting. Filtration is nothing but a harvesting, one of the harvesting after harvesting first step, either centrifugation or filtration to separate the broth and the bacterial mass, correct? Did you all able to correlate or it's completely French and Spanish?
Yes, sir.
One more person, I think. Can you correlate with what we have studied? Because what we have studied fundamentally, I'm trying to make it affirmed and exactly examination based question. If it is antibiotic fermentation, then how you can use both the knowledge together and really score a good marks. Okay, that is my attempt here. So now fermentation, we are going to read it in detail. Somebody else from the class, please read this fermentation. Yeah, this is the first process of step was there. So in fermentation, we discuss about pH temperature. So please read this.
Fermentation. The pH of Chesapeake Dox medium was adjusted between 6 to 7.2. The medium was dispensed in the fermenter. The fresh sample of mycelial suspension of fungus Penicillium Rhizofluvium From the fresh slope of wrapper steep agar, that is caustic dogs medium plus cons plus steep and agar was obtained. The solution was autoclaved for 200 minutes at 120 degrees Celsius at 151 pressure and
Yeah,
fermented
15,
14 days
15 pound. 120 degrees. This is a standard fermentation. Yeah, okay.
for 14 days at 24 degrees Celsius.
Yeah. So after fermentation, you bring it to 24 degrees centigrade and then you put the Penicillium grisophalvus. Is this clear or इसमें कुछ problem लग रहा है? Please
So
take a. Yeah. Somebody has question. Please ask.
yes, so what is corn steep?
CSL. Okay, I'll tell you. I'll go back to the previous one. Yeah. See, here I talked about a pure Zapadox medium, correct? And this is a fermentation medium. Corn steep liquor is this a corn processing when we do. I think corn, correct? And it is the whole processing is done in extraction of that process. We get the byproduct in the corn processing, and in that, whatever that liquor comes out has a lot of carbon, but other cobalt, other salts. See, in Zapadox medium, we have only magnesium, correct? But the magnesium, your question could have been immediately there. Sir, magnesium alone is not sufficient for the life, correct? We need calcium, we need cobalt. This is a big source of cobalt. That's why cobalt came to mind. You need ferrous, correct? Fe, our blood related. So similarly, organism need that, correct? Every life need this kind of thing. So here we add these natural constituents, corn steep liquor. Yeah. And that is why its importance came here. Yeah. When it came to now, we are talking about a fermentation. The basic Zapadox medium plus corn steep we add. Does that become clear to the class? The question was very nice and apt. But how about others? Did you got it? Something clarity, more clarity?
Yes, sir.
Okay, I think go ahead reading. Okay, the next slide. Okay, this is I think should be very, very familiar slide to you all. Just
Yes, sir.
हाँ करेक्ट आई थिंक ये देखो मैं सेपरेट आज मैंने ये आज भेज दिया आप लोगों को आपके ईमेल पे मैं सेपरेट नहीं भेजूंगा ये सिर्फ फर्मेंटर का क्वेश्चन आएगा कि प्लीज ड्रॉ अ फर्मेंटर एंड एक्सप्लेन फर्मेंटर देन यू हैव टू ड्रॉ दिस फर्स्ट ये ड्राइंग कोई डिफिकल्ट नहीं है हाँ मैं खुद कर सकता हूं तो आप लोग तो सब लोग मेरे से अच्छा ही कर सकते हो एनीबडी हैज एनी क्वेश्चन बिकॉज दिस वी हैव स्पेंट द होल क्लास बिकॉज आई स्टिल बिलीव that construction of fermenter is the most important aspect you all should know, understand, and I'm definitely going to ask a question on that. The draw fermenter and parts of label the parts of fermenter and explain the fermenter. So anybody has a question either तो पूछ लो 8 मार्क यही है आपके समझ लो.
Sir, I just wanted to know the two things, the sensory probe and the system monitor.
आपका आवाज टूट रहा है, please ढंग से बोलो.
I'm audible, sir.
You are audible now.
Sir, I just wanted to know the two things, the sensors, probes, and system monitor.
So I think I explained, but I'll again tell you, sensor probes. These sensor probes are, pH electrode. Have you seen pH meter and pH electrodes in your college, school? pH कैसे measure करते हैं पता है?
Sir, we saw the pH meter.
हाँ उसमें see we put something into the beaker. उसके साथ electrode types होते हैं जो pH is ultimately hydrogen concentration we are measuring, correct?
Yes, sir.
Yes, sir.
अच्छा फिर आप लोग कॉलेज आओगे तो मैं एक बार वहां आके आपको दिखाऊंगा कि कैसे है. But that is what a sensor. The electrodes are a sensors which are able to, on the basis of physical and a chemical and electronic basis, they are able to measure the quantity. And in fermenter, what happens is their readouts are completely going directly onto a computer. So that is what a sensor and a probe. So this is I just talked about pH probe. Temperature. अच्छा temperature तो आप सभी लोगों ने अपना भी लिया होगा और कहीं भी देखा होगा. It itself is what? It's a probe, We see that mercury down and then the mercury rises, correct? If this mercury rises, you are seeing it with your eyes. But if I put some kind of mechanism and physics-based mechanism which can measure this rising of mercury and convert it into sensor. So that is how I think pH probes are, temperature probes, correct? कहीं भी temperature लेना है, अभी body का लेना है तो हम thermometer लेते हैं. But suppose if you want to take certain temperature and record for 24 hours, then we have a probes available. ठीक है? That is what a sensor and a probe. Similarly, there is oxygen. These are the three very important aspects for which we have a sensor and a probe. And that is right question, the question for the class, very important question. See, here I have written only sensor probe, but आप जब 8 marks का question होगा, तो आपको sensor probe के अंदर ये लिखना होगा कि what are the sensor probes? They are pH, temperature, ये मैंने क्लास में बहुत बार बताया है. ठीक है, लिख लो आप चाहे तो, ठीक है? I think that was a good question, yeah, Yogesh? Does it answer your question, the question second point?
Yes, sir.
Okay. So I think anybody has any question on this fermenter? I think please continue reading. Whoever is reading, please read.
The system monitor is also kind of same thing over, sir.
Sorry. Acha, system monitor. System monitor is, it is monitoring the whole pressure inside. सबसे ज़्यादा important क्या है? यदि fermenter में, see, it is stirring taking place, growth is taking place. So अंदर pressure बहुत बढ़ता है. Yeah. So pressure से oxygen transfer भी नहीं खराब होती है, and the whole system need to be monitored. So system monitor and sensory probes are slightly different because sensory probes only send the information about pH, temperature, oxygen. But the system monitor does what? It measures this and take the decision on that. ये computer program है. अभी हमको इतना अभी computational जानने की जरूरत नहीं है. But this our artificial intelligence तक जाने की जरूरत नहीं है. Normal computer program writer can do it. If the pH goes up, then you have to, that is if it becomes alkaline, what you need to do is you have to give a shot of acid, correct, to bring it back to 7. And that is being done using system monitor. So they are part of the same, but sensory probes send the information and the system monitor is going to really take a rectification error. यदि temperature बहुत बढ़ जाएगा तो cooling water भेज देगा. Yeah? समझ में आया?
Okay, sir.
I try to make it as simple as possible. Okay, please read this.
Pre-treatment of fermentation broth. The broth is heated.
I think we missed her. I think if anybody else can read it. Okay, I think filter. See, this is like I'll read it. The broth is heated at. Sorry, go ahead.
The period of heating may be short, 5 to 10 minutes of 80 degrees Celsius, having been found to provide a satisfactory increase in filtration.
ठीक है. So filtration में नहीं तो coagulation बहुत होता है. So I think ये part यदि आपको इतना डिटेल में नहीं समझ में आता है कोई बात नहीं. आपने मीडिया प्रिपरेशन, फिर fermentation मीडिया, then fermenter draw कर लिया. The next step is your product is ready inside, correct? The sensory probes and the system monitor tell you that yes, it's a time to harvest. Now 6 days are over. There is no more grisofulvin is been produced. Please start the harvesting process. So this is filtration. Just read this quickly. कितना बाकी है? A lot of things. I think I'll only read it so you can go faster. Otherwise, I'll wait for the whole. See, the filtration can be done using a drum cover with drum filtration. Yeah. So I will just straight away show you the drum filtration. See, can you all see the drum? There is. Can you see one circle?
Yes, sir.
Yes, sir.
हाँ. So वो है ना cross section है. ये ड्रम है. एक्चुअली 3 डायमेंशन में देखोगे तो ये ड्रम है. इसका क्रॉस सेक्शन में दिखा रहा हूं. करेक्ट. सो ये ड्रम व्हाट हैपेंस इज वी कंटिन्यूअसली रोटेट दिस इज अ फिल्ट्रेशन जोन. ओके, फर्स्ट लेट मी एक्सप्लेन यू व्हाट आर दिस इज अ फिल्ट्रेशन जोन. या सो फर्मेंटर ये ड्रम है. ये हमारा फर्मेंटेशन का ब्रॉथ जो हमने कलेक्ट किया 100-200 लीटर, उसके अंदर ये नीचे का पार्ट, द लोअर पार्ट इज रोटेटिंग. करेक्ट. What happens is these are the, can you see the dots in a fermentation zone? they get settled onto this drum. Yeah, because of the suction inside. Can you see the suction inside? ये आप लोग एक बार ध्यान से देखोगे तो आपको समझ में आ जाएगा. अभी मैं बताता हूँ. हाँ, so suction होता है अंदर बीच में, and then it is rotating. What happens is all the fungal cells will get covered onto this. Can you see this brownish belt which is written as a cake?
Yes, sir.
And then
Yes,
here,
sir.
see that cake we are separating. You know what happens with the knife? We are taking away the solid product. This solid product is nothing but the fungal mass, Penicillium grissofulvin and grissofulvus. And through this suction, the liquid part has gone separated. So now we have been able to do, and this filtration is a new type of filtration I showed you. A normal filtration in your mind is just we put a cloth and you can filter. But when you are talking of tens and thousands of liter, actually this is 1 lakh, क्योंकि ग्रिसोफोल्विन is a cheap product. We use a very big fermenter. So इसके अंदर ये ड्रम को रोटेट करके try to understand. I know it is not that easy. If you would have been in here real life, तो मैं कुछ आपको ड्रम दिखा के करता. But try to understand in next class. You ask question if you don't understand. Okay? Because you know you will be having the next class, and last time I overshot it. I don't want to do that. So then the next step is we have got now, as I told you, this is water insoluble product. So we do the extraction. So the extraction is done in a cold acetone, correct? And acetone is an organic liquid. So something which is not so soluble, whole grissofulvin get dissolved into acetone. The extraction with the cold acetone may be carried out with the efficiency of 75. इतना ज्यादा आपको जरूरत नहीं है कितनी efficiency और क्या. Just say that acetone extraction is done and the whole thing is been taken out 95 to 99. But just for some of you who are interested, I'll tell you, the more the efficiently you extract out the product, the more economical you are. Yeah, because that much wastage is less. So first step is filtration, the second step is extraction, the third step in this is decolorization. So color is color of an extract can be improved by addition of a calcium hydroxide. Yeah, calcium hydro in grissofulvin production we add calcium hydroxide after this filter extraction, because extraction acetone extraction में जो आता है वो बिल्कुल it will look like a tar. Yeah, so first thing what you try to remove is the color, unwanted color, which is calcium hydroxide, preferably 30 milligram per liter. This is important. I think you will have to understand in this is a specific with grissofulvin. यहाँ तक किसी को कोई क्वेश्चन है? I think ये definitely this drum you understand, try to understand and go ahead. We can take it again, but extraction बहुत simple है. Decolorization मैंने बताया. And then isolation and separation of the product. The impurities of waxy substance are removed by washing. You see, washing the excess solvent. If you start washing ethyl acetate with water, I think you will be getting rid of other immiscible, and your grissofulvin being non-soluble in water, it will remain in an organic phase. So that is how you are getting rid of unwanted impurities. It can be color, it can be other kind of impurities, and which can be getting rid of. We are not interested in impurity. We are interested in grissofulvin. So we are selectively concentrating the grissofulvin, correct? Hexane, hexane and a petroleum ether here I mentioned contain a high portion of hexane, are generally suitable for this step. If you use this hexane, it can be very useful. Go through this presentation. Some details are not up to little too much of detail at your level, but I think it is interesting to know how people will be. If I am producing grissofulvin, that will give you a full satisfaction. Yeah, and the last step is precipitation and purification. Yeah, it's done by crystallization. Anything which is, if it is in a solvent and if I start evaporating the solvent, what will happen? Its solubility even in solvent will slowly, there is a saturation point. After the saturation point, you give a cold shock and it will immediately start forming a crystals, because cold will even further reduce the solubility very quickly and the crystals of grissofulvins are obtained. Yeah. That is what the whole grissofulvin is. I think, I know that last part was very, very rush, but please ask some question. I'm just leaving 2-3 minutes And in the next class, if any slide number you want, I will focus and explain again.
Sir, I had a question.
Yes.
So what was the work of mycelial cells in history?
Good question. Grisofulvin is an extracellular product, correct? So the mycelium which we get in this, what we try to give a wash. So whatever is attached grisofulvin to this mycelium is we take it out, and the mycelium goes as a bio waste. If you remember your downstream processing, there I said that if the product is not intracellular, immediately we throw away the cell mass, correct?
All right.
It's a good question. Good question. If it would have been yeast, we would have thrown away the yeast which are used for a protein for feed. Human feed में भी आता आजकल starvation जहाँ है वहाँ पे yeast as a protein we give. Then we would have thrown the broth, but that yeast biomass we would have dried and processed and sold it. ठीक है, I think that should be now clear, right? Any other question? You are still 4-5 minutes. Okay. Yeah, go ahead. There is some question. अच्छा, 1 मिनट आप लोग आपके कैमरा ऑन कर देना भाई. जो भी क्वेश्चन है, डोंट वरी, नेक्स्ट क्लास ऑल्सो यू कैन आस्क. हेलो. अच्छा, you all are coming to college in September? No. Okay, ठीक है. Then we'll be having the same way of learning, correct? Yeah. And one more question: does every professor ask you to put on the camera while attendance?
Yes, sir.
Yes, sir.
तो डेफिनेटली, क्योंकि एक क्लास में मेरे को अटेंडेंस डालने में बहुत दिक्कत हुई बिकॉज आई डिडंट आस्क यू टू पुट द कैमरा ऑन. So there is something I don't know what this computer. Anyway, I think that fine. You all will put your camera on, and I would also leave. I think in 5 minutes time your next class is there, right? Okay, then see you all. Yeah, next class.
Thank you, sir.